Well, I'd like to welcome you all to today's talk sponsored by ASEF, the Association of Senior and Emeritus Faculty of the School of Medicine, Perelman School of Medicine. I'm Howard Goldfein, and it's really my great pleasure and honor to introduce today's speaker, Scott Hensley, who's a colleague in the Department of Microbiology in the School of Medicine. Scott was trained first at the University of Delaware, where he received his bachelor's degree, and then came to Penn as a student in cell and molecular biology, where he graduated with a PhD in 2006 under the guidance of Gundi Ertl at the Wistar Institute. After his postdoctoral experience, he was appointed as assistant professor at the Wistar and promoted to associate professor in 2016. In 2016, he migrated over across Spruce Street to our department, the Department of Microbiology. We're very fortunate to have him. Scott has distinguished himself as a leading investigator in the field of influenza immunology, and that's going to be the subject of his talk today. He's been honored as a Burroughs Wellcome Scholar, a competitive and prestigious award. So let's welcome Scott to give his talk.
Okay. Thank you for that nice introduction. I changed my title a little bit. Instead of talking about immune history and influenza susceptibility, I'll get into that a little bit, but I thought I'd make this more general today. And so this is kind of a clunky title, but pretty descriptive of what I'll talk about. Pandemic influenza virus, antigenic drift, and influenza strain mismatch. Okay, so here's the virus. Here's flu viruses, and I'm going to try to keep this pretty general. I'll show some data slides, but we'll try to keep this somewhat general. There are two glycoproteins on this virus. In green is a protein called hemagglutinin. In yellow, these are proteins called neuraminidase. Our lab really focuses on immune responses against the green protein, hemagglutinin. And that's because most neutralizing antibodies against flu, most antibodies that actually prevent the virus from replicating, most of these antibodies target the green protein, hemagglutinin. Now, hemagglutinin could be split into two different regions. The top there is called the head, and the bottom is called the stalk or the stem. Most antibodies that we produce against hemagglutinin target this head domain. And they're really good at preventing infection, because what they do is they bind to this head domain, and they essentially prevent the virus from attaching to cells. We're going to talk a little bit towards the end today about antibodies targeting the stalk, but most of what I will talk to you about are antibodies against the head of hemagglutinin. Now, I think I failed to tell you, but hemagglutinins' main job in life is to attach viruses to cells. And the hemagglutinin molecule does this by binding to sialic acid that is linked to longer sugars on host cells. And you can see in this crystal structure right here, here's sialic acid. Okay? So HA is the attachment factor for viruses to attach to cells. It does it by binding to sialic acid, which is right here. And we mount a whole lot of antibodies against this hemagglutinin protein, and most of these neutralizing antibodies bind up here, and they just prevent the virus from attaching to cells. Okay? Now, here's the problem. There are tons of different hemagglutinin molecules. Okay? Flu viruses, there are many different subtypes of flu, and even within a subtype, there are many different what we call drift variants. And the globular head region of these hemagglutinin proteins in all these different strains, they're very different. And that's why we get infected year to year. So with flu, we have two major problems. One is this problem of pandemic flu viruses jumping from animal reservoirs into humans. And you might not realize this, but all flu strains, their natural reservoir are aquatic birds. Sometimes they jump into pigs and then they jump into humans. Other times they go right from birds into humans. But there's many different, these viruses are very different, okay? So we're going to spend some time talking about this 1918 pandemic strain. This was caused by an H1N1. That is hemagglutinin subtype 1, neuraminidase subtype 1. Before 1918, humans had not really seen an H1N1 virus before, so the population was very immunologically naive. So when this virus entered the human population, it caused a large pandemic. You can see that this virus, after causing a pandemic, didn't just go away. It continued to circulate for 40 years. How do you think it was able to do that? It was able to do that because at that point, the hemagglutinin and the neuraminidase protein, to a certain extent, started to acquire mutations in the globular head domains of these proteins, which abrogated antibody binding. So a lot of people were infected during this 1918 pandemic. They had immunity against it. It was devastating. But now, what did the virus do? It started to change so that it could continue to infect people that had been previously exposed. But you could see, in pretty unpredictable fashions, new subtypes of virus entered the population. So in 1957, an H2N2 virus came in. It was replaced by an H3N2. And our most recent pandemic was by an H1N1 virus in 2009. So there's two main problems to deal with flu. We have a pandemic threat of these new viruses jumping in. And we have this problem of antigenic drift, the process by which these viruses gradually change in these two proteins that I introduced to you, hemagglutinin and neuraminidase. So let's talk about the 1918 pandemic. It's been 100 years since the 1918 pandemic. And Philadelphia was a really special place for the beginning of this pandemic. So I like to show this in my emerging infectious disease class, okay? And shown on the x-axis, it might be a little hard to read here. On the x-axis is year, and on the y-axis is life expectancy. And I asked my students, do you see anything weird here? Right? You can see, it's pretty crazy, right? So you can see life expectancy dropped very sharply during the 1918 pandemic because so many people died of this virus. And not only did a lot of people die of this virus, a lot of young people died of this virus, which I'll get back to in a minute. Now, this is just remarkable. If you look at death rates in the U.S. by month, again, you can see something pretty striking, right? That you usually have some increased deaths in the winter. This goes down. But then look what happened here in September and October of 1918. What happened was the 1918 pandemic, a lot of deaths. So I told you about Philadelphia. So Philadelphia was a very special place for sort of the initiation of the 1918 pandemic in the United States, and I'll go over why that's the case in the next couple slides. But here, this is I don't know exactly where I got this slide, but this is looking at mortality in U.S. cities during this pandemic. And you can see here, this might be a little hard for you to read, but this is September through November, November to February, February to March. And we have cities here. So Philadelphia was really, during the early days of this pandemic, really was the major place, one of the major places in this country where this took off. Okay, and this is one of the events of, this is one of the major events that was associated with this sort of explosion of the 1918 pandemic in Philadelphia. So does anyone recognize this street here? It's Broad Street, right? And this was a parade. This was the Liberty Loan Parade. And so this was a military parade. That's right. And if it was 1918 and if there was a parade in town, guess what? You went to it, right? Because parades are awesome now, but they were even more awesome in 1918. There wasn't, cable TV and internet and stuff. So here's the parade. There are so many people that went to this parade. 200,000 Philadelphians went to this parade. And basically, it was thought that a lot of these soldiers were infected, and people have looked back at this date, which is just over 100 years ago, as a major cause of this great outburst of flu infections in Philadelphia, this commingling of infected people with large crowds. And I tell you, so, I mean, you think our healthcare system's stressed now, and it is, but back then in 1918, if you're interested in this 1918 pandemic and the history in Philadelphia, I urge you to read Penn Medicine magazine just had an article on this, and it describes how there was an abandoned hospital on Cherry Street. This is right as they were building the Ben Franklin Parkway. And so they were getting ready to condemn this old hospital, and then they put a resident in charge. They were like, you know, we'll open up this hospital just to house people infected with this 1918 virus, and they actually put a resident, someone that was super young, in their 20s or 30s, to head up the operation, and he tells stories of you know, going to bed at night and waking up and having, you know, a dozen individuals dead every morning during this 1918 pandemic. You can see here, and this is in Kansas, right? This is a temporary hospital. There's a lot of these neat pictures if you go around online. I was just in Iowa. Iowa had this this huge sort of basketball pavilion where they converted this into sort of a temporary hospital. So you can imagine that you know, our healthcare system was maxed out at this point. And, you know, simple things that we take for granted now. We didn't have antibiotics. Okay, antibiotics obviously wouldn't do much for a virus infection, but most flu infections, most flu deaths during the 1918 pandemic were associated with a secondary bacterial infection. Okay, so a lot has changed in 100 years. Okay, so this is something that's really interesting. Something my lab actually studies is the age distribution of different pandemic flu strains. And so the 1918 pandemic was strange in that young people were the most affected. Okay, during a seasonal flu outbreak, what we typically have are the very young, our kids tend to get very sick from flu, and older individuals tend to get sick from flu because their immune system is going downhill. In the 1918 pandemic, what we saw on the x-axis are age here. So you can see these bars. This is just what? Percentage of deaths. You can see a lot of these 20 to 30-year-olds were dying. You really seldomly see that with flu. And this is something that we have been studying quite a bit, as I just mentioned. So how do you study the 1918 virus? Well, you need to have the virus, right? And so this is one of my favorite stories of all time. So Jeff Taubenberger, who is at the NIH, got together with a historian named Joan Holton. And Joan had written books about the 1918 pandemic, and in particular, he had this one chapter on how this virus just devastated this town in Alaska. It was a tribal town, I guess, and basically everybody in the town died. 95% of the town died. And what they did was they dug sort of into the permafrost and just made this mass grave of essentially everyone in town. And so Jeff had this idea of, well, let's just try to dig these people up. And since it's so cold, maybe we can actually get viral RNA that had remained dormant for whatever it was, 80 or 90 years. And sure enough, they went up to Alaska and they got permission from the tribal leaders. This is still sort of a protected land, and they exhumed the bodies and got the nucleic acid enough to then recreate the virus. And so how did they recreate the virus? So for flu, we have something called a reverse genetic system. It's really nice to be able to work in this system because we have plasmids that encode each one of the flu genes or gene segments. And if we throw in, there's only 8 of these gene segments for flu, and so if we throw them all into a cell, what we get live virus. So all you really need to know in the case of flu is the nucleic acid sequence, and very rapidly we can generate virus based on that nucleic acid sequence. So with that said, you can imagine this was a big storm when people, when Jeff Taubenberger and colleagues wanted to resurrect the 1918 virus, right? I don't know if you guys remember this controversy. This was maybe 2004, 2005. But there were a lot of prominent scientists that sort of weighed in on this. And David Baltimore wrote a really nice sort of opinion piece that I think helped get this published in 2004, 2005. You can argue that you think about the timing here. This is right after 9/11, so you can imagine that giving the sequence of a pandemic virus would be a bad thing, that people could recreate this virus. It could end up in the wrong hands and be a terrorist type of thing. You could also argue that there's a lot of basic science to be learned by understanding the virus that caused the worst pandemic in our known history. And so, one way or the other, this sequence was published, and we've learned a heck of a lot about this virus. And I'll argue that it has helped us understand our susceptibility to new pandemic strains today. What did we learn? Well, the virus was deadly in animals. I don't want to go through all the data. Suffice to say, this is body weight and survival, and these are different animals. So animals are surviving against different viruses, but here are animals that got the 1918 virus. There was a lot of genetic experiments completed to try to understand what genetic determinants made this virus so lethal. I'll point out one really nice study by Peter Palese, who's going to be here in the spring to give a seminar. He found that a single residue in that hemagglutinin protein that I began the seminar talking to you about, a single residue here at residue 190 increased pathogenicity of a seasonal virus if you made it look like this 1918 virus. And this is complicated and something you probably don't want to chew on right now as you're having your lunch. But basically, the single residue in the hemagglutinin made this virus attach to human cells differently. Okay? And birds and humans have different sugar compositions on the outside of our cells. And something got cut off there. And so what was going to be shown here is basically how this hemagglutinin protein engages these different sugars. And one thing we learned from the 1918 pandemic is if there's a single residue swap in this hemagglutinin protein, and if it can engage a human receptor in just the right way, that it's one way for these avian type viruses to adapt to human hosts. We've learned a lot too, and I don't think I'm going to talk about it too much today, but we've been studying the 2009 pandemic, and we've learned that much like the Spanish flu, middle-aged individuals seem to be differentially affected by this virus, and we've actually determined that susceptibility to this strain is really dependent on which viruses we have seen in childhood. So, for example, if you have been infected with a 1977, 1978 virus, we have found different susceptibilities to different H1N1 strains, these contemporary H1N1 strains, compared to someone that was infected, say, in 1990. And what we're doing now is we're really trying to understand this side of the equation. I told you that there's this sort of weird age distribution here where middle-aged people had preferentially a lot of disease to this virus. And our hypothesis is that is because they had some unique exposures here in the 1800s. And what we're trying to do now is we're actually trying to take a page from Jeff Taubenberger's book, where we are trying to resurrect viruses from the 1890s. And how would we do that? Well, as it turns out, we have a very old hospital here, and there are actually autopsy samples that date back as early as like 1890. And pathologists, I don't know if there's any pathologists here, but you guys don't throw anything out. And so I've gone to all this stuff is in Roxborough, and I went up to Roxborough a few months ago, and I probably should have put some pictures in here because this is an interesting thing. But there are just dozens of these boxes, and if you had an autopsy from like 1960s, 1970s, we can pretty much find the box of tissue that corresponds to that particular autopsy report. 1890s, not so easy. So. I'm trying to recruit a grad student to look through these boxes. I actually have a postdoc that might start with this. But the idea is if we could find the lung tissue, and it's been sitting in Roxborough, Lord knows, where is it? It used to be by 30th Street Station, and then rent got high. Now it's out towards Roxborough. So who knows what this material has gone through. But if we can isolate the 1918 virus, from Alaska, then I think we should be able to get enough nucleic acid out of some of these lung sections. And if we were able to do that, then the experiment would be to then throw this into animals, where we would then infect animals with these older viruses and then come in with the 1918 virus to see if that would increase their pathogenicity. So this is like a big dream, and 5 years from now, maybe we'll have that. Okay, so what I want to spend the rest of the time talking about is not pandemic strains, but rather seasonal flu viruses, which we have to deal with every year. And I would argue are probably a bigger problem than pandemic viruses. Because as I pointed out early in my talk, you get these pandemics, they cause a great amount of disease for a year or two, but then they stay with us for up to 40 or 50 years. And again, the reason they do, the way they could do this is by undergoing anagenic change. This is a phylogenetic tree that shows the genetic diversity of H3 viruses, in this case just from 2006 to present. Each one of these circles is the sequence of a single virus. And what you can see is that there is constant change. And unlike a lot of viruses, what you have is you have this very directional change. At some points, you can see, I'll just give you an example, 2012, you did have sort of local competition between these three clades, but you can see there's always one clade of virus that sort of wins out. Now, the challenge from a vaccine standpoint, how do we vaccinate? I'll go back one slide. What we vaccinate, we have currently one of these H3 viruses, one of these H1 viruses, and we have one or two influenza B viruses in our vaccine, antigens from these viruses. So what we do from a vaccination standpoint is we're sequencing these viruses constantly, and we're trying to guess. We're trying to make an educated guess about what's going to circulate in the future. And today I'm going to show you an example of where we had a bad guess, which was, I guess, during this time period, this 2014-15 year. And then I'm going to show you a problem with our contemporary viruses where we have a pretty good idea what's going to circulate, but we still fail to make good vaccines against these viruses. Everywhere you see an X is where these vaccines have been updated. So you can see we don't update the strain every year, but we update it pretty frequently, and it's kind of a guessing game. Here is a slide that demonstrates how long it takes to make flu vaccines. And we're about to get into the season here in Philadelphia in the Northern Hemisphere. And we need to choose what's going to be in the 2019-2020 vaccine, so next year's vaccine. We have to choose that in February because it takes us a while to make what we call a high-growth reassortment, a virus that can grow in chicken eggs, which is typically used to make vaccines. You got to make them, you got to package them, you got to put them on planes, and you have to vaccinate the population 9 months later. So it takes about 9 months to actually, from when we choose these vaccine strains to when we actually get vaccinated, and that's a whole lot of time for the virus to change. So again, the outline of the rest of my talk, sometimes we choose the wrong strain, and sometimes the vaccine actually changes as it is produced. Okay, so I don't have many slides on choosing the wrong strain. This is something my lab does do quite a bit, studying each of these mutations as they arise in nature over time. But I think this slide nicely illustrates it. This is from a small paper that we had in 2015. And in this flu year, you can see that there are these, what is this? These are different viruses, different clades of viruses. So you kind of had these green viruses, you had the black viruses, the gray viruses, and the red viruses. And you can see that sort of early in the year, this is the summer, so these are viruses coming from the southern hemisphere. There was kind of a mixture. But then as we went through the northern hemisphere season, really, we had these guys dominate. So what do you think the vaccine strain was this year? I kind of already set it up by the title, right? It was the green one. Right? So that was a terrible decision. And to no fault of the WHO and the CDC, but it's complicated. And you remember, this vaccine was actually chosen in February. So at that point, these green viruses were more prominent. So sometimes the virus just sort of evolves faster than we can make the vaccine. And so we're, in some ways, always sort of playing catch up. Okay, but something that we have focused on quite a bit over the last year or two is sometimes we actually choose the right vaccine strain, but that the strain actually changes as we are producing the vaccine. And so I want to introduce H3N2 viruses to you. H3N2 viruses have been around since 1968. These viruses actually cause the most disease on a seasonal basis. And something happened during this 2014-2015 season. During this 2014-15 season, it was a bad vaccine mismatch. That's when we were looking at the phylogenetic tree. I was trying to find this on the phylogenetic tree. It was a bad mismatch, and it was a bad vaccine mismatch because of this single residue in hemagglutinin changed from a K to a T. And you might be like, well, how could a single residue do that on HA, on hemagglutinin? The way it did it was that this was not just some ordinary change in hemagglutinin. This was a K to T that led to the introduction of a new glycosylation site on hemagglutinin. So this is the hemagglutinin structure looking at the side of it, and this is looking from the top down. So if you're sort of an antibody, you're kind of approaching it from here. And shown in black are glycosylation sites. Okay, so not, your host proteins are glycosylated, whatever. Viral proteins are glycosylated as well. And you can see hemagglutinin has a lot of these black glycosylation sites on it. In 14-15, this blue glycosylation site was added. Viruses that circulated before this did not have this glycosylation site, so it rapidly emerged. And the reason that the addition of glycosylation sites can have such a major antigenic change is that they're big. You can think of these almost as like umbrellas. This is classic in the case of HIV. HIV has tons of glycosylation sites on the envelope protein, and that's one reason that HIV is hard. It's very difficult to generate a neutralizing antibody response against HIV. So we have a single residue change here. We didn't see it coming. It emerged sort of as our vaccines were being produced. As the season began, we all kind of, bit our fingernails because we knew that the vaccine was going to be poorly matched. And sure enough, it was a bad mismatch. But then, 2 years later, we solved the problem because we now updated the vaccine to have one of these blue strains. But we have this problem of egg adaptation. So how do we actually make flu vaccines? What we do is we take these viruses, we inject them into chicken eggs, okay? This is kind of an old technology. And the virus then replicates in the allantoic membrane of these chicken eggs. And tons of virus is basically secreted into this allantoic fluid. And people in my lab do this. I used to do it a lot when I was actually pipetting. And it's a very cool thing. You can just put a needle into the allantoic membrane and suck up this virus. And then it's inactivated, and the hemagglutinin proteins are enriched. And that is basically what's in your vaccine. 90% of our vaccine supply is made in a chicken egg. The problem of the chicken egg is that human viruses, they engage sialic acid, which is the receptor for flu viruses to bind to. They engage sialic acids that look different. There are different sialic acids in humans compared to chicken eggs. So you take a virus that is really good at attaching to human cells by engaging a specific type of sialic acid. Now you throw it into chicken eggs. What happens? It acquires mutations for it to bind strongly to these chicken cells. Now, in the case of what I'm telling you, we updated the vaccine strain to this vaccine strain called A Hong Kong, blah, 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 2014. This is a virus that was isolated from the nose of someone living in Hong Kong in 2014. And it's matched to what's circulating actually today, pretty well matched. But then after doing this process of egg adapting this strain so it can grow to high titers, then we get this A Hong Kong strain that has this reassortment number next to it. This had the T160. This has the glycosylation site. And then after passaging in eggs, look what happened. We had this reversion. Okay, so we chose the right vaccine strain, T160. This has the glycan intact, but now upon growing this thing in eggs, it has basically reverted to look like a virus that circulated in 2011, 2012. Okay, glycosylation matches the circulating strain, and then here we don't have this glycosylation. This is the slide I like to show people. So you can do this on your phone now. So this is me, and I don't know if, I'm not really at a good angle here, but I don't have a lot of hair on top. It's kind of like mostly on the side. So you can think of me as sort of the vaccine strain, the current vaccine strain. And you can think of hair as sort of the glycosylation pattern. So I have some hair on the side. I kind of have this bald head. And you can imagine if you raised antibodies against me, and if the hair would prevent the antibodies from binding, you'd probably have a lot of antibodies up top, but they would fail to recognize the strain that actually circulated. This is an example of the 16-17 season, but this is also true from last year, 17-18, and it's going to be true again this year if we have H3N2 viruses. So we've done experiments in ferrets studying antibody responses elicited against these vaccines. I don't think I have those data in here, but basically antibodies against the egg-adapted strain failed to bind to the circulating strains. But we also did these human experiments. So I told you that most of the vaccine in our country is produced in chicken eggs, and that has a mutation, this reversion mutation. We have some cell-grown vaccine, but that also has this reversion as well. There is about 5% of our vaccine supply is produced through a baculovirus-based recombinant system. Okay, these are baculoviruses infect insect cells, and you can make recombinant proteins in insect cells. And this is just this is great because you're not sort of reliant on actually expanding the virus in different host cells. So you don't have this problem of the virus changing. It's basically just a recombinant protein that has the wild type sequence. So we actually looked at humans. These are humans that were vaccinated at the University of Rochester. And here's our little key here. And basically, this is looking at a fold rise in antibody titer to the circulating strain. Okay? And each one of these circles is a human. And you can see that this vaccine called FluBlock, this is the insect cell one that actually has the wild type sequence, elicits a very strong response to the wild type strain, and we get much weaker responses when we looked at vaccines that were produced in chicken eggs or in cell culture. And if you look at antibody responses against the egg-adapted strain, you really don't see too much seroconversion, and that's because these vaccines look like 2011 viruses, 2012 viruses, and we all already have titers against those to begin with, so we don't really have this rise. Okay, so this probably goes without saying. So, should we still get the flu vaccine? Of course we should, right? So, yes, so even in years of vaccine mismatch, the vaccine prevents against severe illness, hospitalization, and deaths. So, if we go back to this example, we have this bald, sort of semi-bald vaccine, a full set of hair on the virus. You get a situation like this where even if you get vaccinated, you might have most of your antibody responses against an epitope, in this case with this egg adaptation, that fail to recognize. But what we know is that you still get some of these antibody responses, although they're subdominant, that they can still prevent severe disease. So when you hear that vaccine effectiveness is 0% last year, that is really looking at how well the vaccine prevented primary care visits. Okay, and it doesn't really take into account how well the vaccine actually prevented hospitalizations and really severe deaths. In the United States, we only track flu infection or flu deaths in children, and so it's hard to really quantify that based on adults. And so this has always been said, but I don't think there's so much, there's not too many studies that have actually looked directly at this until this study that just came out a few months ago. So I think this is a great study if you're interested from folks at the CDC and others that have actually shown that that you have decreased severity even in times of vaccine mismatch. We're working with Arnold Monto's group at the University of Michigan, where during this 14-15 season, despite the fact that the vaccine effectiveness was essentially 0.5%, in a hospitalization study there, we've actually seen increased something like 40 or 50% effectiveness. So get your flu vaccine. Get all your kids vaccinated, your grandkids vaccinated. That goes without being, that's obvious. Okay, so I only have maybe 2 or 3 more data slides. Can we make better flu vaccines? So my lab, 90% of my lab is really studying antibody responses and where antibodies target different viruses and what types of exposures lead to different antibody footprints and whatnot. But we have a small fraction of my lab that is actually working on bona fide vaccine research. Can we make a universal flu vaccine? And so let's return to this slide. And what I've been telling you about mostly is antibodies to hemagglutinin. Again, these antibodies bind to the globular head of hemagglutinin, and they prevent the virus from attaching. And again, these antibodies are very strain-specific because this is precisely where the virus tends to acquire changes. So there's a big interest in the community in trying to make vaccines that target this lower area called the hemagglutinin stalk. And the stalk is attractive because it's very conserved among different flu strains. The problem is that if you just do a serological survey of different individuals, most of us have very low antibody titers against the stalk. It seems to be immunosubdominant. So how do you make a vaccine against the stalk? I should just say that these antibodies don't block attachment. They work through other mechanisms, such as keeping the hemagglutinin locked together so it can't undergo conformational changes that are required as part of the viral life cycle. A lot of these antibodies also work through different mechanisms that involve Fc engagement of innate immune cells. Okay, so what about this stalk? We're certainly not the only group looking at this. There's a number at VRC at the Vaccine Research Center at NIH has some really promising stalk vaccines, as well as Mount Sinai's group. Okay, so, I think this is nice. This is a structure here of hemagglutinin, and this shows an antibody at this point, just the Fab of an antibody binding to the globular head of HA. Here is an antibody binding to the stalk. What do I want you to take away from this is antibodies are big. I think that's something that I didn't realize until I start looking at these structures. So you can imagine that, there's a lot of different mutations here that can prevent this head binding antibody from actually binding. There are a number of mutations down here in the stalk that can prevent these antibodies from binding, but this part of the virus is really refractory to change because it just needs to be in just the right conformation in order to do what it needs to do, which is undergo this really complex conformational change. So the antibody footprints are big here as well, but again, these you don't see much variation because this part of the virus cannot change as much. Okay, so here is what we've been working with. We've been working with the modified RNA vaccine, and I like the title here, the Influenza Vaccine of the Future. And this isn't something that I've invented. This is Drew Weissman's work. Drew Weissman's been working on this for a number of years here at Penn, and he's developed these modified RNAs as vaccine antigens to a number of different vaccines. And we've just worked on the flu component with him. And I have no stake in this game at all, but this vaccine is just incredible. I'll show you some data, and it really does look promising. So what did Drew do? What Drew has is he has this lipid, and then he has nucleic acid expressing whatever antigen you want to express. In this case, the hemagglutinin protein. And then what Drew did was their nucleic acid is very immunostimulatory. If you put nucleic acid in the wrong place in your cells don't like that. It's a danger signal. Your cells can sense the RNA and say, let's shut down translation. Let's initiate a strong innate immune response that basically will shut a virus down. So what Drew did was he took these RNAs and modified them in a way that these sensors inside our cells cannot recognize the RNA. And you might be thinking to yourself, well, wow, aren't you trying to make a vaccine? Don't you want to have a strong immune response? Well, if you need to have antigen, you need to have protein in order to get a vaccine type of response. And so without modifying the RNA, you basically just get no production of, in this case, hemagglutinin, and no antigen in order for your immune system to see. So this is really the trick, is to modify the nucleotides in a way that they cannot be sensed, and then they are packaged in these nanoparticles, these lipid-based nanoparticles. And at the time I was at Wistar, and Drew sent me an email saying, we have some interesting data looking at this with a number of antigens. Would you like to work on flu with us? And we did a couple initial experiments, and it was just like, wow. And let me show you a couple of those wow experiments. So here is one of these experiments. So this was something we published in JEM earlier this year, with Drew being the senior author of this study. And so you can see here, this is HI titer. So this is antibodies, in this case, against the globular head of HA. And this is what a conventional vaccine looks like. And this is time post-vaccination. In this case, this is a mouse, but we've done this now in rabbits and we've done this in ferrets. This is a mouse. So you can see that this a very long-lived response by this conventional vaccine. But look at this guy, 5,000 compared to 80. I mean, these are titers, 5,000 for what we call an HI titer against flu. This is just something that we don't see, okay? And then, so now we can then we can infect these mice, in this case, with viruses. And in this case, we're infecting with the same virus, the matched virus that was encoded by this vaccine. And you can see in this case, this is looking at weight loss. So animals that didn't get a vaccine, well, they lost weight and they died. And then animals either getting this conventional vaccine or Drew Weissman's magical vaccine, they survived. And you can look at this and say, well, it's not that much better than the conventional vaccine. But what happens now when we have mismatches? And that's the key. So this is a paper where we've had the joint effort here, more joint, that just came out in Nature Communications. And this is the same kind of study. So here we are immunizing with Drew's vaccine that encodes the hemagglutinin from this virus called PR8. And you can see that at day 28 and 238, you have very high titers. This is measuring antibodies against the globular head of HA. But now what we've done is we've looked at antibodies against the lower part of HA, against the stalk. This is a little complicated if you're not used to thinking about ELISAs, but this is basically looking at two different time points. In black is day 28, and red is day 238 after vaccination. These are antibodies that can react to the total hemagglutinin protein. So this is just diluting out the serum from these mice. You could say you have a big signal, and then you could dilute it out, it goes down. But now this is shown here in this panel. These are antibodies specifically targeting this hemagglutinin stalk because we coated these ELISA plates with antigen that only had that domain intact. And what you can see is, well, it's less, right? You don't have all of your antibodies against the stalk. This is a lower signal compared to here. But you have a substantial signal of antibodies binding, and they actually increase over time. So this is the response at day 28, and they go up by 238. Now, here's the key. Now, what happens when we do the challenges? Well, here is a challenge where the vaccine actually matches 100% to what we challenge animals with. This is another mouse experiment. And you can see here that you get, here is the, in black is the conventional vaccine, and red is the modified RNA vaccine, 100% protection with both of these vaccines. But now, what happens now when we have a drifted strain? So in this case, the vaccine is something called A California 2009. This is a drifted strain where the HA is very different. The hemagglutinin is very different. You can see that the conventional vaccine does not protect at all. These animals rapidly lose weight and they die. Now, if you test this drifted virus in animals that receive this modified RNA vaccine, well, they lose some weight because these antibodies are not sterilizing. They don't just gum up the virus and prevent infection, but what they do is they help clean up the infection through these non-neutralizing mechanisms, and these animals can regain weight and go on to live a happy life in the animal facility. So we have done this in, like I said, in mice, in ferrets, in rabbits. We're doing some non-human primate experiments, and Drew Weissman is really sort of the driver of sort of the commercialization of this, and he plans human studies in the not so far future. Okie doke. So what have I told you today? So pandemic flu strains pose a constant risk, and I would argue. So our conventional vaccines afford essentially zero protection against these, and we need to develop new universal flu vaccines that can protect. Right now, the government is stockpiling certain vaccines against H5 and against H7, and these quickly become outdated because these viruses are undergoing change slower, but still undergoing antigenic change in these animal reservoirs. Flu viruses are constantly evolving in humans, and for that reason, we have to update these vaccine strains. And it's not a perfect process, right? So sometimes the viruses evolve, and then they're different than what we immunize against. And sometimes as the vaccine itself is immunized or as it's manufactured, you get these mismatches. This is something that Wharton should figure out, right? This is, it is clear if you have a vaccine that is made through the recombinant protein system, it will be better compared to these egg-grown vaccines. And now what we have to do is figure out how to convince companies to switch over. Because you can imagine this is a complicated thing. There's completely different equipment that's made or used to make these two different vaccines, but it really is. This is a business thing, right? This is starting to get a little bit out of the realm of science. The company that makes that baculovirus hemagglutinin it's a small company called Protein Sciences, 5% of the market, and guess who just bought them? Sanofi, who is the biggest producer of egg-based vaccines. And when we saw that, we said, "Oh man, are they buying it just to bury it?" And I don't think so. I think they're actually good people in Sanofi. I think they are very forward thinking. And so here's a big company that now has acquired this recombinant protein-based system, and it would be really good for them to have some incentive to expand that program. Everyone in my lab tries to get the recombinant protein vaccine, and they have a hard time finding it. Okay, so even when there's vaccine mismatches, you should still get vaccinated, right? And so, at the end of the day, okay, yeah, there should be this push towards using these vaccines that are already appear to be a little bit better. What we really need is a large-scale vaccine effectiveness study. You can't, I showed you data from my grad student that was looking at like 50 people per group. You're not going to make, huge policy changes based on that. What we need is thousands of people enrolled where they get different vaccine types. We track them over a couple years to see if there really is superior protection. And once we have that, I think we can then convince some of the manufacturers to change what they're doing. But in the meantime, one way or the other, we need to push new universal flu vaccines forward. And NIH seems to be committed to this, and we're happy to be playing a small part there. Okay, so with that, most of the funding for my studies have come from the NIH as well as the Burroughs Wellcome. Sears is a Center of Excellence grant that I'm affiliated with, which we are going to go in for our own grant here to make a Sears Center at Penn in 2020. I try to mention people as I went through. Here are the grad students who've already left me, and a number of grad students and postdocs in my lab. And the modified RNA vaccine work here at Penn with Drew has been a lot of fun, kind of pushing us into different directions that we're not used to, and that's been a good collaboration. So with that, I think we have 5 minutes or so if there's some questions. Thank you very much. Yeah.
Back to 1918 for a minute.
Okay.
You mentioned briefly a hypothesis that might explain why so many young adults died in the 1918 flu compared to seasonal flu. And you suggested that it might have to do with some unique exposure, viral exposure 20 or 30 years earlier. Mm-hmm. I can certainly see, it makes sense that would explain why the 20 or 30-year-olds would be dying and the kids would not be dying, but how would that explain why the older people were not dying? Because presumably they would have had that same exposure 20 or 30 years ago.
Yeah, it's a good question, and I wish I had a different slide in here, but I'm just going to go back to the timeline of what we know. Where are we? So, I mean, there are always, we don't know the history. But the hypothesis, we don't know the history here, but the hypothesis would be that older folks were exposed to something that had protective epitopes conserved between the 1918 virus and something, I don't know what the age thing was, but say it was 40 years. Well, that would be 1880. So viruses that circulated, 1870s, 1980s would actually confer protection against this. So it's not that, prior exposures are a good thing.
I see.
They can be not great. So in this case, their prior exposures, this is something, we haven't really worked with the 1918 virus in my lab, it's BSL-4, but we've worked a lot with this 2009. And what we found is that there are some exposures that occur between 1977 and 1985. I mean, it's like a very defined window, which is amazing because we have all this sequence data from back then. And those exposures protected individuals during this 2009 pandemic.
So I should have asked the opposite question. Why would it?
Why would it be detrimental?
Or no? Why would?
The older people be protected.
Yeah, but so.
Why would the middle-aged
kids not be?
Yeah.
Kids wouldn't have had that exposure.
a lot of hand waving here because we don't know, but the hypothesis is that that these middle-aged individuals, which, you subtract, I guess the age group is probably in their 20s or 30s, so you subtract 20 years from this, and 1890-ish, that...
There was a pandemic in 1889, 1890.
Yeah, and so, it was thought to be an H3 virus, but there was not a whole lot known about this. We don't know exactly what the strain is, but the idea would be that if your immune system first saw that virus, that you would have an immune response so focused on that subtype of virus, which is in a completely different family as this, and that now when you're exposed against this, that you would recall memory responses against this, and that would come at the cost of generating a de novo, a new response, and that you're almost better off not even seeing the virus, and that the kids would still be affected, but not. And there's some data that supports this. I've given a talk tomorrow at Vanderbilt, or in 2 days, and I'm going to be talking about this because if we look at, it's hard to do that experiment because we don't know exactly what virus is circulating. That's why we want to go back and try to get them out of freezers, or not freezers, just blocks, autopsy blocks. But what we can look at viruses like H5 and H7, which are sort of causing local outbreaks in parts of the world right now, which are sort of the highest pandemic threat type viruses. And the observation is that whether you saw an H3 virus or an H1 virus in childhood, you have different susceptibility to these H5 and H7. And it works completely as you would think immunologically. So if you had this as a kid, you are protected against an H7 virus, but more susceptible against the H5. And that makes sense with what we know about how these viruses are in the different families of virus. Whereas if you had this as a kid, you are more protected against H5 and more susceptible to H7. I have to cross my eyes when I think about that because it's probably just, that probably doesn't, the specifics aren't important. But it's amazing if you think about it. I mean, like, how do you get susceptibles in the population? I mean, you're just moving this way. You have kids are born and people die, right? And like this, we're just going forward. There's no going backwards. So, I mean, the population, I think we work with Josh Plotkin a little bit in the biology department. He gives a lecture in my emerging infectious disease class on this. And, with flu, the way you get susceptibles is, birth rates increase, death rates can increase, and then but then the virus can change, and the virus can make someone that was exposed. They can throw them into the susceptible compartment. But so what is our pandemic going to be? I think we can learn from these immune histories and sort of predict what our next pandemic strain would be. And the argument would be that this H1 type of immunity is falling off because these people are dying. And that the H3 exposed people are becoming more populated.
So this is probably another seminar, but in broad strokes, can you see any parallels between what's happened with the flu virus and the HIV virus?
Yeah, well, there are a lot of...
Aside from the mutation.
Yeah, well, the high mutation rate, my HIV colleagues like to show a phylogenetic tree of HIV next to a phylogenetic tree of flu and say this is like how much evolution happened in one person in HIV, and this is...
But there's no virus now. I mean, that's what I mean. There's been a lot of viruses for vaccines for flu, but there's no vaccine for HIV.
Yeah.
So
What's the deal with HIV? There are a lot of parallels. So with HIV, there are some neutralizing antibodies against HIV, and they target sort of these conserved epitopes that are in the HIV virus, just like I'm telling you that there's this conserved epitope in the flu stalk. What's similar about those conserved antibodies? Well, they're kind of weird antibodies. They're antibodies that have very long regions that can kind of fit into these grooves and kind of get into weird places. So they're weird antibodies. And they are, in both the case of flu as well as HIV, these antibodies are thought to be induced by sequential exposures. What happens during a sequential exposure? You get exposed once to an antigen, you have a B cell response. Now you get hit with a virus that's a little different. What happens is B cells can undergo something called somatic hypermutation, where they actually change the B cell receptor to change a little bit. So now you can recognize that drifted strain. Now you get hit with another antigen that's different. Oh, I'm going to change a little bit more. Now you're going to hit, and so in the case of flu, that might take 50 years because you got to get exposed to flu viruses over a course of 50 years. And your antibodies that get induced when you're 3 years of age, you know, those B cells that get initiated at 3 years of age, well, they get tickled every 5 or 10 years, and they change a bit to recognize these drifted strains. In the case of HIV, it happens in a couple years. It happens in the course of, because in the case of HIV, you're constantly getting these antigenically drifted strains exposed, you know, from your reservoir. You have immune escape and essential exposure. So in the case of HIV, one thought is to try to, you can't take one vaccine antigen, you can't take one HIV antigen and try to make a vaccine. Now what people are doing are trying to figure out what type of antigen do you need to initiate a broadly neutralizing response, and what kind of boost do you need to get that to differentiate. So right now, you know, there are 5 different, you can imagine a scenario where you do 5 different immunizations with these antigenically distinct HIV strains to try to narrow the antibody response to these conserved epitopes. Same kind of thing people are thinking about with flu. There are some parallels. Yeah.
Yeah, so you make a convincing case that this new way of manufacturing flu vaccines is going to be much better, and the trick is going to be to convince manufacturers to adopt it. But, you know, you've got the FDA there in the middle. Mm-hmm. They're extremely cautious about manufacturing. Are people at the FDA working with you and people who are doing this so that they're not confronted with something brand new all of a sudden?
Yeah, so it's a good question. So with the protein-based vaccines, those are already approved and used in humans. They're just a small fraction of what's used in humans because there's not enough production capability to scale them up. So they're already approved. In that case, it's just a matter of convincing companies to increase the production. And from the FDA standpoint, you need a large vaccine effectiveness study to try to label that as being more effective. You can't just say, Seth Zos, PNAS paper, blah, 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 right? I mean, it's a small study that we've done. So as far as Drew's vaccine, I do think that there are a number of companies that are working on this, and they're actively engaged with the FDA, these nucleic acid-based vaccines. There's a company in Cambridge, Massachusetts, Moderna, where I think there's some like intellectual property disputes between Penn and them. But they've already initiated human trials, and they're kind of moving forward. So I'm really not involved with that, but Drew is sort of the guy here on campus that's really savvy as far as what to do pushing that forward. Okay, thank you.